I do this. And so I'm also quite happy to have Andrew here today, and also the topic Turing patterns. I don't know who doesn't know Turing patterns and all the different beautiful math that goes behind it. But from my point of view, Turing patterns are also, as much as you want to use them, you're also scared of them because you don't know what do they do. And in terms of development, there's a lot of information out there. So the neural tube, there's a lot of morphogens, how you do the dorsal, like the ventral axis, all the axes of the body seems to be run by these morphogens, and some sort of Turing patterns is involved with them, but we don't know enough, at least in humans. But where it puzzles me the most is when you go not from development, but from regeneration. I always wonder, like, okay, so we got all these patterns and morphogens directing the digits, let's say, but what happens when you chop one off? How do they know, where do they go? Where did they start? Does the pattern just start fresh? I mean, those are just interesting questions to think about. I just wanted to kind of promote discussion, but I'm sure Andrew's going to give you a lot more to think about. So Andrew, the floor is yours. And thank you all for being here.
Yeah, thank you very much. Sorry, I didn't introduce myself because you'll have to endure, right, me speaking for such a long time. I should say, right, if this is coming through, I always tell people, particularly at groups like this, so I'm quite a theoretician and not a very good biologist. And so if you get bored at any time during the talk, if you Google my name and you find this website, you can find some sort of fun pictures and simulations and things. So as an example, reaction diffusion systems, Turing patterns on, say, growing domains and manifolds and various things. I always just like pointing that out. Right, so let me bring this up. So I put here my Twitter handle, not because I'm sort of looking for people to follow me, but to tell you I'm legally blind. So particularly if you go to the Society of Mathematical Biology in Heidelberg, I hope to see some of you there. Or if I bump into you at a meeting, there's no way I'll recognize you, and it takes me a while to learn voices. The other thing is because I'm at a UK university, I put math here to tell people I'm an American, right, as opposed to maths. So right, okay, let me start talking. I think as well, I'm happy if people interrupt me as we go through this. This, I hope, is fairly informal, and I hope is somewhat useful. So the topic, as Maria said, is sort of pattern formation and understanding things like digits and things like zebrafish skin and all of these sort of wonderful examples of patterns. Before I get into this, I really want to put some caveats. So as I said, I'm quite a theoretical person, so I'm not necessarily saying I'm a very good theorist, but this talk and quite a lot of what my expertise is in mathematical studies and reasonably theoretical studies of quite simple models. To sort of justify this or maybe make a case for why this might be useful for you, I want to read this quote from Richard Levin's in this fantastic paper on the strategy of model building and population biology, who more or less makes the argument, right, that you have this horrendously complicated natural state, and we have kind of feeble human minds, and so it's very difficult in general to satisfy all of these criterias of understanding, having a general theory, having a realistic theory, and to be able to understand and control biological phenomenon. And really, there is no simple solution. There is no real solution to this. And so what Richard Levins argues for, and what I think is quite important, especially in these sort of interdisciplinary groups, is to have a large mixed strategy. So that means many people doing many different things, even from very competing philosophies or perspectives, each kind of contributing their own piece. I think this is quite important, and I'll come back to this at the end. So this is my sort of mea culpa, that the rest, or a bit of this talk at least, will be somewhat theoretical, and I'll try to focus on the things that are relevant for biological modeling. Another thing, really, if you take nothing away from this talk, it's that if you haven't read Turing's paper, you should at least give it a read, because it was published in the Philosophical Transactions of the Royal Society B, meaning it was aimed at biologists and chemists of the time. And I think, I mean, again, I come from a biased perspective, but I think he really does a good job trying to highlight what he's getting at and what the modeling means without delving too much into sort of arcane mathematical formalisms. Each of the sections, I've put a couple here, has kind of, I think, a nice intuitive picture of what he's trying to get at in terms of meaningful biology. So I'll come back to this in a minute. If you are interested in these topics, I will be very narcissistic in this talk and mostly talk about my own work and the things that I've been involved in, which will be a subset of this blue paper here, which is a review paper on this sort of linear stability analysis, which will be most of this talk. But more broadly, we put together a philosophical transactions B issue, which again, you can find on my website, that has all of these other papers written with some biologists, some physicists, some chemists, and quite a few mathematicians. That the point was really to review the state of the field. And so I think we did an okay job, or I should say, I think the other contributors did a great job in putting this all together. So I'm often not very good at sort of thanking people for work and things, but I try to put everyone's name where they are. So this in particular was done with Eamon Gaffney, Philip Maney, and Václav Klika. But this issue, we hope, is useful for particularly graduate students and newcomers to the field to get some sense of what the sort of broad picture really is. Right, so for the rest of this, I will talk now more about my own perspective, my own fairly limited view of these phenomena and the sort of progress that we've been able to make, and then the sort of challenges of where this goes. So let me first state some examples of biological patterns that I've been interested in and that I've worked with some success in some cases and quite a lot of failure in other cases in trying to understand. So in fact, the first phenomena, the first example of a pattern which got me into this was doing a postdoc with Eamon Gaffney, trying to understand mouse whiskers. And this work has still not appeared because we're still taking quite a long time to poke at it, but we think we've made some progress. But the point about mouse whiskers that relates it to Turing patterns is that it is a periodic pattern, which is sort of, as I'll get to, the sort of key thing that comes out of a reaction diffusion model or theory of a reaction diffusion mechanism, but it's not just a periodic pattern. So there's a kind of macro scale variation. There's a size and a wavelength modulation of the follicles that become the roots of the mouse whiskers as you go down the apex of the mouse's snout. And then there's also this structure that you really see these hairs appear here in an almost predefined region in these rows. And these rows don't have periodic hexagonal packing. They actually have something that looks more complicated. And so this process was to try, or modeling for this process, I should say, there's quite a lot of good experimental work, was to try and unpick all of these different features. Where does the size and wavelength modulation come from? Where does this orientation into rows come from? So other examples of this kind of macro scale patterning are work that hasn't gone anywhere, right, because I haven't done much, but some fantastic experimental work by Anna Dobritsa, and then theoretical work by Adriana Dawes, who I think is here, on these really beautiful structures. So their work was on these dark apertures, but I really am obsessed with these pictures because you can see there's sort of periodic patterning underlying both these dark regions, these apertures, but also these bright green regions, which is an exene called protein on the surface of pollen. Other work with collaborators at UCLA has looked at bat teeth and has looked at trying to understand bats, which are not a model organism, by sampling different taxa and trying to understand the role of a plausible reaction diffusion mechanism and understanding the size and spacing of bat teeth. Again, the theme for all of this is that you have some periodic pattern, but there's macroscopic variation, or there's kind of structural bits to it which make it not a simple periodic pattern. More recently, we have work with Dennis Heaton's group on human fingerprints and then analogs in mice. And the idea again here was to try and see if a sort of reaction diffusion chemical process could underlie the sort of variations we observe in fingerprints, and then the experimental work on mice to sort of probe perturbations of these gene regulatory networks. I won't go through any of this detail, quite a lot of histology, quite a lot of experimental work, but I hope this appears soon. And again, these are structures where there is some sort of underlying periodic pattern, but you do have macroscopic things like arch, loop, and whorl patterns. You have things happening at the boundaries of your fingerprints, and you have a lot of interesting structure in these patterns. Finally, I like to mention some work we did with Microsoft Research, who at the time in 2020 were quite keen on synthetically developing biological systems to exhibit Turing patterns. So they had bacteria, which they genetically engineered to use quorum sensing molecules to signal between different colonies of bacteria to try and mimic a pattern. And this is quite hard. So my contribution to this is a very small thing, but I enjoy this because I can tell people that when I went to Microsoft Research, they had a single wet lab biologist who had a lab full of robots. So they were genetically engineering bacteria with robots, which sounds sort of like the beginning of a science fiction story. But anyway, it was very cool. It was a very cool project, the sort of things that they were working on. And I think they had several papers. And there are other groups, I should say. Again, I'm talking from my personal experience. There's quite a lot of interest in doing this sort of synthetic engineering. And I think there's quite a lot of value in understanding the more complicated, in my opinion, cases of in vitro or mammalian biology by trying to mimic it synthetically with simpler organisms like bacteria. Right, these are just sort of random examples of periodic patterns. So let me sort of tell you what a Turing pattern is, where it comes from, and how, as a mathematician, I understand it. And I'll try to relate this back to the biology. So again, I think it's quite valuable to take a look at Turing's paper at some point. He essentially thinks of two chemical species and two equations like this, which I'll define in a minute. And what comes out of this, particularly at a computational level nowadays, are things like spots and stripes and really interesting spatial structures that kind of spontaneously emerge from this mechanism. So let me tell you a little about the mechanism. If we assume that my two chemical species are well mixed, spatially homogeneous in something like a chemostat, the classical sort of systems biology or chemical engineering approach to this is to use something like the law of mass action, to have a system of ordinary differential equations representing the rate of change of the concentration of two populations. So I should say there's a lot of work on stochastic versions of this. There are a lot of work on more thermodynamically sensitive versions of these kinds of models, but at a sort of coarse level, as long as the assumptions of well mixed and reasonably dense number of molecules per volume are satisfied, then you can model biological systems, things like gene regulatory networks, via these ordinary differential equations. The point of Turing's work from a mathematical point of view is, what if I don't have this in a well-mixed chemostat? What if I allow these molecules to move around? So there are many different kinds of movement, but the simplest might be to add a term of diffusion, right? So now the rate of change of the density of U is a function of the concentrations due to the local chemical reactions plus some diffusion term. And for simplicity, I'll start with the sort of one-dimensional domain, and I'll assume that my molecules don't leave the domain, right? So X is in some space, I have some spatial line that I'm thinking of, and I don't let things leave. And we want to ask, can this model here have different steady state, particularly behavior, than this model up here? And one thing you know is that if I think of a spatially homogeneous equilibrium, so a flat state where the concentrations are the same everywhere in space, then that equilibrium must be the same between these two models, right? It has to satisfy that the nonlinear kinetics, f of u and v, are equal to g of u and v are equal to 0 for both models. So these homogeneous steady states are the same in both models. And you can then ask, does their stability change if I have this diffusion term or if I change these diffusion parameters? And this is the sort of, again, mathematical point of view on Turing's work. So the way that I understand these things, or the way that I teach these things, in order, right, for me to write exam questions for mathematicians, is to focus on this procedure of linear stability analysis. And I'll come back to what this means later. But the idea is, I think of starting at this homogeneous equilibrium, perturbing it with these capital letters, so thinking of some small kick to the system, and then asking how does this kick to the system evolve? So you wind up with now two linear partial differential equations for the growth rates of these perturbations to see whether or not perturbations grow or decay that look like this. This matrix J here is exactly the Jacobian that you would get for the ODE model stability analysis. So that Jacobian says everything about the reaction kinetics and everything about the reaction mechanism, but doesn't incorporate the spatial bit. And the spatial bit is given by this diffusion matrix and then these partial derivatives with respect to x. So this is, again, just the diffusion term in each species. And then the question is, right, and then we can solve this to determine something about these growth rates. So you can formalize this and justify this in a few ways. When I give this talk to pure mathematicians, this is where they get grumpy, but you can plug in this sort of classical physics ansatz, substitute this in, and get out a quadratic equation for the growth rates. And what this means is that for every k, for every sort of cosine wave, every sort of kind of spatial perturbation or Fourier mode, you have a growth rate associated to it. And that growth rate satisfies this quadratic equation. And I should say this is the part where things for the undergraduates that I teach, I try to encourage them that the rest of anything one might do with this is just studying the solutions of quadratic equations. So in principle, the mathematics is sort of at the high school level. There are details, there are things that you have to keep track of, but I hope, right, if you at least believe that I can plug this in and get out this thing, that the analysis of that quadratic equation is something that one can work out fairly easily on pen and paper. A Turing instability is exactly when k is 0. So I have a homogeneous perturbation. This would be a perturbation for my ODE system. If the growth rates to such perturbations have a negative real part so that they don't grow in time, then a Turing instability is when a growth rate to some spatial perturbation, so some cosine mode, grows in time. And this is the sort of mathematical way of representing that. So again, there are details here, but I think most of the mathematics in deriving this quadratic and understanding the sort of basic conditions for Turing instability is reasonably well understood. And again, I would encourage you to take a look at Turing's paper for more details, or James Murray's book. Right, so I've given you this sort of random fact that if I perturb the system in a homogeneous way and it doesn't move, it sort of returns to an equilibrium state, but I perturb it in a spatial way, this somehow gives rise to patterns. Well, you actually need a more sophisticated analysis to show this formally, but it emerges because of something like a pitchfork bifurcation. So what is this? This is if I have a single species that satisfies this equation, then this bifurcation can be drawn this way. So this u star equals 0 is a homogeneous steady state, right, satisfying this ODE, and it's stable as long as p is negative. So if I start at 0 and I give it a kick in either a positive or a negative direction, it will just return to that state. But as soon as I tweak the system parameters so that p is positive, that homogeneous state, or u star equals 0, is no longer stable. And instead what you have are these other two equilibria, and you go to one or the other depending on exactly where you started, depending on exactly your initial conditions. So roughly speaking, there are details here. This is what happens in reaction diffusion systems. If I assume there's some parameter at which your homogeneous steady state is stable for the parameter being negative, and then if the parameter crosses some threshold, you have something like this pitchfork bifurcation. Now, the mathematics for the sort of nonlinear bifurcations here tend to be quite complicated, but the point is that this linear stability analysis is a decent proxy for what's called a supercritical Turing instability, which is what I've drawn here. There are other cases like subcritical instabilities, and there are more detailed nonlinear things, which I won't talk about. So the point of this slide, the point of this bit, is to justify my statement here at the bottom that if I satisfy these conditions, which again is just studying the roots of quadratic equations, then I expect to see patterns. I expect to see some spatial structure spontaneously emerge from some small perturbation or small movement of some homogeneous state. I start with chemical species that are at a flat concentration, and I expect them to grow into something which has an emergent spatial structure. So by analyzing these quadratic equations, this is the part where the undergraduates grumble, you can wind up with these conditions. And by the way, I should define this. F sub u means the derivative of the growth rate of F, the reactions involving the species u, with respect to u evaluated at the equilibrium. So this F sub u roughly represents the interaction of the chemical species u with itself. So if F sub u is positive, we tend to refer to that as a self-activator. And if G sub V is negative, we tend to refer to that as a self-inhibitor. And this is what gives you the terminology of activator-inhibitor. And you can get out from these algebraic conditions, again, under some technical details, you can get out these kind of useful biological facts. So that the only sorts of systems that exhibit these Turing-type instabilities are ones where you have short-range activation, long-range inhibition. Again, the details here are not too difficult to work out, but this is a general statement. So we're not assuming that we start with a particular form of the model. We're not assuming we know anything about the chemistry other than we're restricting ourselves to two species. And you immediately get out this general insight that in order to have these Turing patterns, you have to have systems of a particular form. And this is something that I'm most interested in before sort of any other kinds of insights, so these general mathematical insights that you can say things about large classes of models, or in principle, large classes of physical systems. Okay, so why is this surprising? Some of this is that mathematically, if we think of a scalar system, we think of a single species model, this kind of instability can't happen. One way of seeing that is if we do exactly the same procedure for a single scalar equation, in order to assume that the system was stable in the absence of diffusion, we needed the derivative of this nonlinear function at the steady state to be negative, and then diffusion is only going to add this bit, and this bit is a positive number with a minus sign, so the growth rates for spatial modes in the scalar case are always smaller. You can do more than this. You can in fact prove general theorems that inhomogeneous steady states have to be simple for scalar reaction diffusion systems. So what this says is that a single chemical species, even if it interacts in a complicated way, so for example, if it's a bistable system, it will not admit complicated spatial structures. So even in the case of bistability, you expect at most one turning point between the two equilibria, et cetera, et cetera. Physically, this also sort of makes sense. If you put food coloring in water, the action, the thing I'm adding, is diffusion, and diffusion will cause the concentration of something like food coloring to be uniform or homogeneous everywhere. Okay, so I want to mention this bit of Turing's paper where he says this model is a simplification, an idealization, and consequently a falsification. So Turing had this really humble opinion of his work, I think, that it really was the first step, and he was aware of quite a lot of complexity, quite a lot of mechanics that are neglected in thinking about this, quite a lot of other important physical processes in developing these kind of simple models. And I want to quote again from Richard Levin's paper where he says that any truth from modeling comes from the intersection of independent lies. And I think that this is a difficult thing for us to grapple with, is how limited our mathematical models are. But that said, there's been a tremendous amount of work. So here I'm giving the number of a section of Turing's paper and then an example of a handful of papers, but really almost every section of his paper highlighted some phenomenon in these mathematical models, and this has been extended and written about by many people. In particular, I want to mention the use of computers. So in Turing's paper in figure 2, he had a very crude estimate of one of these patterns, but nowadays there are many, many online tools that will solve reaction diffusion models in a web browser or on your phone. The sort of computation of these models is now very easy and extremely accessible. So there's been a tremendous amount of work in all of these different ways. And quite a lot of this is in generalizing the basic ideas. Right, so as an example, you can formalize a lot of this for things that are much, much more general than two-species reaction diffusion systems. So you can think about manifolds or surfaces. You can think about graphs or networks. If you think of, say, chemostats connected by tubes, and you can do a general analysis of these systems under some technical assumptions. So here I'm thinking of a sort of generic reaction transport system. I have a technical assumption on what I'm calling an operator L. This is not terribly important. And then I can plug in the same perturbation ansatz and determine essentially the same equation for lambda. It won't be quadratic if I have more than two species. It might be cubic, or it might involve other things. And the sign of these rho k's is kind of important. But the point of this sort of general picture is that all of the geometry and all of the effects of the transport appear in this term rho k, and all of the effects of the reactions and the kinetics of your interacting species appear in this term involving J, and these are sort of cleanly separated. And you can do exactly what what the sort of algebraic conditions I showed you before do, and show that in more dimensional species, you need an activatory subsystem, which in some sense is short-range activation, an inhibitory subsystem, which in some sense is long-range activation. So you can kind of cement this insight in a much broader class of mathematical models using these sort of linear stability analysis tools. Let me give you a few examples where I've personally written papers and looked at sort of toy mathematical models of these things. So we've looked at things like spheres and tori. These are nice mathematical abstractions. We've done lots of simulations. We've applied this linear stability analysis. We've looked at models of cross diffusion. So this is where you replace your pure diffusion with these terms like this. As an example, this includes things like chemotaxis or things like directed motion. And we also looked at inertia, right? So really diffusion has an infinite speed of propagation when thought about this way with these sort of continuum models. And we sort of followed the same textbook analysis that I showed you on the previous slides. And we've looked at, I have a decent amount of work in population ecology, which uses the same ideas, but we looked at the case of directed motion, right? So all of these sort of equations, all of these different models are the same sort of thing. The analysis looks very much the same as in Turing's paper. It's generalized, it's a bit more mathematically sophisticated, but the ideas are identically the same. And again, the general insights of things like short-range activation, long-range inhibition hold true for most of these models. And then we've also looked at cases of things like Stokes flow. So if I submerge reaction diffusion system in a slowly moving flow or really a low Reynolds number flow, you can do this sort of linear stability analysis. And more importantly, I think at least, we have powerful computational simulations now. And so you can really explore three-dimensional structures in these flows, right? And you can really see what happens. So in all of these cases, right, again, these are all fairly theoretical, you follow more or less the recipe that was in Turing's paper. What I want to spend a bit of time on shortly is mentioning where this recipe doesn't work out and where it becomes more difficult to get at these general insights, right? So one of these, there's quite a...
Can I interrupt you? Sorry, I just have one quick question, just to sort of like tie this a little more to development. So
Yes.
when you say that it's more than one species, I think of them as genes. Like, so you have inhibitors and activators. When you're doing this network, have you ever had one gene to be both an inhibitor and an activator, or is there a rule each one has to be specifically, because it just in development, it gets a little complicated. Inhibitor of one can be an activator of another. So I wasn't sure if there's a rule to stick to.
Yes, that's a really good point. So when I say activator inhibitor, there are two bits to this. One of them, I'm not actually, so this is a sort of classical misconception with the terminology. When I say that the first species U had to be an activator, so I said that means this F sub U is positive. This is only the species interaction with itself. And when I say that V is an inhibitor, that means that G sub V is negative, and that means that's only the interaction of the inhibitor with itself. In fact, the interaction of the activator on the inhibitor or the inhibitor on the activator can be either. So there are cross kinetics or pure kinetics, depending on if the activator also activates the inhibitor or vice versa. In the multi-species case, there are two things that are important. One is that we have a way of classifying if a subsystem is self-activatory or self-inhibitory. And again, that sort of is a loose way of capturing what a whole subsystem is doing. But the other bit that you mentioned is that it's not always the case that something is an activator even of itself all the time. And this is because you can have different steady states. So all of this is thinking about a single spatially homogeneous equilibrium, but as the system moves away from that, you might be in a different state. And so this sort of linearized Jacobian doesn't capture that, right? You get different numbers in this matrix J, so they may change sign. So one system may be an activator inhibitor system for some regions in the parameters of phase space, but it can be quite different in other regions. Does that sort of make sense?
Yeah, no, I was more interested to see if there was a strict rule, like because networks, at least when you start looking at gene interaction networks, genes interact in different ways depending who's their activator, inhibitor, or what in space. I just want to make sure that this theory captures that, or we have to stick to, at least I always found the Turing, you always needed one activator and one inhibitor, and there was some rule to define it as Turing, and I wasn't sure what happened in multi-species. So thank you.
Yeah, you can generalize it under, again, some technical assumptions, and there are some limitations, but in particular, this very last equation I have written here is the same. And from that equation, again, it becomes not a quadratic equation, it becomes an nth order equation, but you can make some general statements about the kinds of systems that admit Turing instabilities. And you typically need subsystems to follow loosely this short-range activator, long-range inhibitor. But exactly what that means for a subsystem to be self-activatory, right, is a bit technical. Right, so let me mention now things which are more difficult from the mathematical point of view, but probably push things into a more realistic setting, which is something that Turing knew himself, which is to say that you don't really have this spontaneous pattern formation from a homogeneous equilibrium, right? A fertilized embryo at no point that I'm aware of is ever perfectly spatially homogeneous. A loose way of capturing that is to just stick some spatial dependence into the system. So here I have highlighted in blue some X, and to say that the reaction kinetics depend on some spatial variable. This is sort of only valid if I think of time scales where whatever caused a pre-pattern is done and the pre-pattern is fixed. And this X more or less captures this effect of this pre-pattern. So I'll come back to time scales later. This has been talked about by quite a lot of people in terms of combining Turing-type pattern formation with other ideas, so positional information, or a solution to that problem known as the French flag model. So you can think of, for example, if I look at these leopard spots, you can see that there are two regions, there are sort of roughly two spatial scales that stand out visually. One are these sort of broad regions where you have some dark area, and then within that dark area, you have sort of dappled patterns on the interior. And this is fairly easy to capture with spatial heterogeneity and with this sort of combining ideas if I had some, say, reaction diffusion mechanism or other patterning mechanism, which created large spots, and then I have a secondary mechanism initiate, and this causes patterning within that. And this has been used by many people. So I got into this because this is what we think of now with some experimental data in terms of spatial modulation of Turing patterns due to gradients of things like retinoic acid and gradients of BMP production. And so I won't talk too much about the mouse whiskers. What I want to go back to is a kind of linear instability analysis. Again, this will be technical, but I hope at least on the last part of this, I can try and get at some general insight that comes from this. So here we assumed that if you had a pre-pattern, the pre-pattern was sufficiently slowly varying. So the time scales of diffusion and length, and the t here is the time scale of reaction kinetics, were such that the pre-pattern was slowly varying with respect to, say, the pattern wavelength that we're looking for. And doing some mathematical analysis, which I won't go through in detail, you get these horrific expressions. This, right, does not look like e to the lambda t times the cosine. This looks much more complicated. But from this, with some work, you can deduce localized Turing conditions. And so these are identically the same conditions that were on the first slide I showed you with these algebraic conditions, but they now depend on x, right? And the reason they depend on x is I'm linearizing about something like an approximate steady state. So it's no longer spatially homogeneous. It's a steady state satisfying my sort of heterogeneous reaction kinetics set equal to 0, and here this Jacobian pulls out an x. So there are mathematical difficulties in studying these systems, but we think anyway that we've justified these kind of local Turing conditions. And what do these look like? Here, this black dashed line is my heterogeneous steady state. So this is what would happen to my reaction diffusion system if I was locally at every point equal to the homogeneous steady state according to, say, the parameters. And this red line is more or less where your classical Turing conditions would predict pattern formation, and this is what we see. And then we show that this matches well as you decrease this epsilon. So in effect, you decrease the patterning wavelength with respect to the sort of domain size or with respect to the scale of the heterogeneity. And so this is somewhat right. When I give this talk and there's a physicist in the room, they say, well, this is obvious, you've stuck an epsilon there. And then I give this talk and there are pure mathematicians in the room, they say, well, you haven't actually shown this is all formal asymptotics. So it's sort of in this boundary that we've shown something which I think makes intuitive sense, that locally, if a system can form Turing patterns, it will. There are details that you can have interactions here, and I'll get to that in a minute. But using this sort of local theory, Thomas Woolley and Eamon Gaffney and I went about applying this theory. So one way of doing this, or one way of visualizing this, is to design Turing systems. So here I take a Turing system and I use Alan Turing space as a spatial heterogeneity in some reaction diffusion system, and I get out somewhere where I match the local heterogeneous pre-pattern and somewhere where I have spontaneous pattern formation. We can design more complicated systems using mathematics we've not shown to be true, but we think are true according to this local theory. And so in these systems, you see there's a white region, sort of gray region, and then a darker region. And then the resulting patterns from the reaction diffusion system are spots, stripes, and then matching a complex pre-pattern. So the point of this work was to say that, well, with this intuition of localized patterning, it's reasonably easy to design these kinds of systems and thereby to design Turing patterns to match quite complicated spatial patterns. Now, it's important to note that that's a very phenomenological point of view, that I can match some complicated pattern, and really this is, I think, a limitation of this theory, because you need more than that, right? Just because I can match any pattern doesn't mean that the mechanistic origin of that pattern really is a Turing-type mechanism with a pre-pattern, right? There's quite a lot of phenomenology, which I'll get to at the end, that you have to be careful using these mechanisms to state what the cause of a pattern is. This is a technical bit that we think we can extend this to higher dimensions, which is difficult, or we think it's true in higher dimensions. We think this is also true for chemotaxis and cross diffusion, again, with a suitable notion of a base state and a suitable notion of a small scaling. As an example of what I mean by phenomenology, someone sent me this picture of zebroids, right? Or zonkies, which is this organism. So this is particularly interesting from a developmental point of view, because Turing's initial insight, or the initial view of these reaction diffusion mechanisms, said that when you have large spatial regions, you expect more intricate patterns, or you expect patterning to occur only when your domain length was sufficiently large. But in these zebroids, we actually see the opposite, right? We see there's some sort of patterning occurring in the legs, but none occurring in the body, even though there's in principle more space for patterning to occur in the body. So again, this is not a mechanistic explanation, but I responded to my colleague with some simulations on a horse where I just assumed I had a pre-pattern that allowed Turing patterning in the legs and didn't allow it in the body. Again, this is not an actual theory of what's actually causing this boundary, but it's suggesting that this is consistent with a pre-pattern or consistent with some kind of developmental boundary. But in some sense, I view this as a limitation of these models, that they aren't actually telling us. We have to do much more interrogative work to determine if that's the real mechanism. Finally, I should say that we've also done a bit of work on cases where this sort of intuitive local theory breaks down. So spatial heterogeneity can cause nonlinear things to happen, and so it can cause moving spots. So I more or less discovered this by doing simulations of these mouse whiskers and then seeing the mouse whisker follicles move, which we didn't expect to have happen in a real developing embryo. And so we sort of narrowed this down and determined some of the reason that this is occurring. But the point here is that we don't fully understand this. We don't fully understand the cases where this sort of simple, localized theory of pre-patterning makes sense, and cases where spatial heterogeneity, some pre-pattern, can cause more complicated things that you didn't predict, you didn't put into the model. So I use this to sell, particularly to mathematicians, that there's quite a lot of interesting work to be done here. Two other examples of these sort of non-classical dispersion relations. Here by dispersion relation, I mean spatial modes which grow, or you expect a pattern of a certain wavelength, are that we've looked at growing domains, as I said, and there are some details, but roughly speaking, you can get out something like those algebraic conditions, although they depend now on time, and so there's some analysis, some details you have to do to understand them. What you get out of the theory is not quite so simple. You don't get out these very easy general principles, but you learn at least something of what the effect of growth and dilution is. And then we recently had a master's student work on gene expression time delays. And so this goes back to work of Nick Monk and Eamon Gaffney in the mid-2000s, saying that if you really have a genetic signal entering a cell, well, there's typically some time for that to induce gene expression in that cell to where it can actually produce signaling proteins and send these out to respond. So you can model this with fixed delays, which is what the mid-2000s work did. And so our student then extended this and generalized this to distributed delays, and this is now in the archive. And the takeaway here is that we think that distributed delay, representing sort of stochastic time in gene expression, doesn't really matter. It doesn't really change the results from having a fixed time delay, but that fixed time delays have a severe impact on the theory. And so this is a kind of interesting question where a more realistic model of the reaction diffusion process actually makes the system less likely to form patterns. And at least from my point of view, I think this is still an open question of what really is the effect of the time it takes for gene expression to occur. Right, I won't go into that in any detail. The point is that you can get something out of this equation, but this thing here looks really messy, so it is not a quadratic equation. It's much more difficult to get general insights, and so a lot of the students' work was numerical, although we think we have a few good arguments in there. Okay, right, I've given you a bit of idiosyncratic mathematical extensions to Turing's system, but now let me talk about what we don't know, or where we can go from this. So I think that the work that I and other collaborators and many other people have done has extended our theory, but particularly our two species reaction diffusion systems are in stark contrast of simplicity compared to real gene regulatory networks, compared to real biological phenomena. even theoretically, we still have deep challenges. And so one of these challenges, which was known to George Oster, I think 34 years ago, is that if I assume there's another process giving periodic patterning, so here this was the Murray-Oster mechanochemical model. So if I think of a mechanical disturbance, like the buckling of a beam, I also can get out things like fixed wavelength patterns. I can still get out periodic patterns. And this is unfortunate if we can't really probe mechanistic details, because all of these models give the same kinds of periodic patterns. And so which is the actual mechanistic explanation of the pattern we observe in biology? So from my point of view, I can group some of these challenges, and there are others, into the fact that these reaction diffusion mechanisms are really caricatures. As you say, you don't, I mean, really in development, you never have, that I've seen, a simple system of, say, Wnt and BMP, of just an activator and just an inhibitor interacting with each other and not touching anything else. Even if we think of just the signaling dynamics, we're several orders of magnitude off of the total number of molecular species, but really things like metabolism and things like cell movement, et cetera, also play a role. There's rarely these sort of elegant, simple processes in biology, and I'll explain a bit of where that comes from. But in particular, there isn't typically a separation of timescales for things like patterning processes. There isn't a sort of simple explanation in many, many cases, from my point of view. And there are quite a lot of other things which I didn't even mention. So developmental biology's real stochasticity, not just in terms of initial conditions, but also in terms of the dynamics themselves. There are many other physical processes. I mentioned mechanics, but there's also cell movement. There's also growth. And here I don't mean growth in terms of just some simplified view of a domain growing, but I mean of cells undergoing mitosis. Theoretically, these nonlinearities, I've given you a perspective. This linear instability analysis perspective is sort of agnostic to the nonlinearities, but we know that these nonlinearities matter a lot. And I don't know of any developmental system where we know exactly what these nonlinearities look like. So they're almost always, from what I've seen, caricatures of the real biochemistry, the real genetic signaling. And there are a lot of limitations in studying models purely computationally or purely from this sort of vague theoretical sort of general perspective, but without respect to particular details. So we can learn a lot, I think, by combining these things. And this is exactly why it's a problem of capturing exactly the right level of detail. So most of our theories of hierarchical patterning, like the one I presented to you here in terms of pre-patterns, assume a real separation of timescales between subsequent patterning events and between different physical processes. And we rarely have this in a very clean or nice way. And I should mention that I think that really looking at philosophy is also quite important, really looking at how we approach these questions and how we think about these phenomena. So some suggestions, right? These are all very idiosyncratic and very personal. As I said, I strongly endorse this idea of diverse mixed strategies, talking with many different people, even competing in some sense, right, of different perspectives. I think this is what we need to do to really understand these harder biological questions of form and function, of where things come from. I think it's important to really try and be specific about what the question we're asking is. So there was a good recent paper by James Sharp on the French flag model and positional information more generally, talking about rephrasing our theoretical biology in terms of questions. There's an older blog post by Artem Kaznichev, which talks about how you can sort of sell a bit of work to a biologist by saying, well, I know the biology is oversimplified, but mathematicians find this interesting, and then sell the same bit of work to mathematicians by saying, well, I know the mathematics isn't interesting, but the biologists find this interesting. And this is sort of something that I fall into sometimes, and I try to avoid, of really making progress in answering a field relevant question. And I think having clear questions is a good way of doing that. I think that this sort of field, right, this group in particular has become very interdisciplinary, but I think it could be more so. I think there's an advantage to talking with science, technology, and society people in sociology and philosophy. I think in order to really formulate and operationalize these questions in a clear way, we can learn a bit from the humanities, but we can also learn on the technical level, people who have overcome similar challenges. So recently I've been chatting a lot with fluid dynamicists and astrophysicists about how they understand spatial structure formation in these fields. And then finally, something that really should be its own talk is to remember that we're all sort of humans, right? We're all sort of doing our own research, poking at our own problems. And I think it's incredibly important to supervise people and be kind to them and to mentor people and to really contribute to these communities like this one here, right? I really, really appreciate Maria's work in sort of forming this group and trying to cultivate a community, a culture of really helping each other out. So I am where I am. I've been able to do the work that I've done because I've been incredibly privileged in how people have treated me. And I think it's always a good reminder that as we're doing our science, right, this is important to not forget the people involved. With that, I'm happy to take any questions or really just have a discussion.
Thank you. This was wonderful. Thank you for that nice comment too. I hope we could help each other. Does anybody have a question to start? I mean, I could, oh, there's a few already. So, Fred, do you want to go or
yeah, in my thinking about echinoderms and pattern formation and stuff, I ended up feeling that there were pre-patterns that were being followed, but in reflection, I'm not sure that pre-patterns are an answer. They're just the same question said differently.
Yeah, I think this is really, really difficult, right? So almost all of the biological work I've been involved in has been a case of iterating with, here's what the experimental results say, here's histology of gene expression, and then me doing a simulation saying, oh, well, I can get that, I can obtain that. But you have to really constrain the models, because if you don't constrain what you're doing, you're thinking in terms of both the verbal operational model of a pre-pattern or not, and then the mathematical formalization of that, it's very easy to match whatever pattern you want, right, to get whatever answer you're sort of looking for. And it's very, very difficult not to be able to do this. It's very hard to I think, use mathematical models like these ones, which are very capable of producing lots of things, particularly pre-patterning. I find pre-patterning very difficult. Yeah, exactly as you say, right? Is that the real mechanism or not? And I'm almost never convinced, right? I'm quite skeptical, despite doing primarily pre-patterning work, right?
Thank you.
Thank you,
This is great talk, and I have a super philosophical question to you. People all over are speaking about new physics, new mass. How would you guide the new mass, considering the difficulties that you just presented? Where to look for new mass? Because mass is enormous. And you use some topologies, some tori, and I saw some spiral too. And I love all these objects, but how to exploit or converge focus on where the new mass should be?
Yes, so I have no answers, right? I have no idea what we should do. I think something that has been useful is to just explore, right? So there's a book by Renee Thom on morphogenesis.
Oh, Renee Thom.
Right, so Renee Thom was very, very much a pure mathematician, very formal, very abstract. He did a lot of work on something called differential topology, And he wrote a book on morphogenesis that almost no one has heard of that does morphogenesis, right? Because it was unlike, I think, Turing's work. It was written. I mean, he was humble about the statements, but he was trying to understand form and function, and going back to these sort of classical questions of on form and growth, right? Darcy Thompson's work. But the actual technical content of this book did not engage with the modern understanding of developmental biology. It was very much an abstract exercise, very much a purely theoretical exercise. I think these things are good, and it's worth doing. So I spend a lot of time talking to PDE theorists and to quantum physicists and this sort of thing. And I think it's incredibly valuable to have discussion groups like this one that involve some of these people if they're interested. But at some point you need someone to really cross boundaries, to really translate between one area and another. And this isn't always successful, and this isn't always easy. But I think encouraging it, right? Or having groups like this is, I think, the way that we make progress, right? Sorry, that was...
So machine learning, AI, and dynamical systems, they have some potential here?
Yes, absolutely. So there's been quite a lot of work on doing exactly what you suggest, which is looking at machine learning and dynamical systems and trying to understand general principles which are present in the data. So these ideas actually go back to older work by Takens and others on looking at, right, so there are these very nice dynamical systems tools where you can look purely at a time series and then get out some kind of model, get out some kind of differential equations or some kind of mathematical model that emerges that sort of matches your time series. And I view a lot of the work in machine learning as being able to do this, being able to produce plausible models. The bit where I think we need more work is the connection between that and mechanistic models with physics and particularly with developmental biologists who understand the physical phenomena and who can say, well, yes, this model is consistent, this makes sense with our sort of working hypotheses of how the biology really works, right? But that means you need like 4 different people in the room, right? You need quite a lot of people who are willing to speak in many different languages, and this can be quite difficult.
So what's the reference to Taken?
There's this old work on time series reconstruction for attractors. So this was work emerging out of Fitzhugh-Nagumo and Hodgkin-Huxley kinds of dynamics. You study things like an attractor of a dynamical system. And then I think within a couple decades, it was obvious to say, well, what if I don't know the differential equation? I don't know what the model is, but I have the time series. Can I construct what a sort of model that represents what this time series looks like? And so there's this stuff on time delay embedding, right? The application is very different. The application is mostly to things like chaotic dynamical systems. But it's an old idea of trying to marry data with sort of classical physics, sort of dynamical.
This is a book by Taken?
No, it was a series of papers. If you Google time delay embedding, or if you look actually at Maryland College Park, their mathematics department, I think, has quite a lot of people who have been doing this sort of thing for a couple decades. It's not especially relevant to development. It was mostly a comment just that there have been people in dynamical systems working on exactly this, looking at data and trying to understand where it came from point of view.
Good, thanks.
Thanks, Maria. Thanks for putting this together. And thanks, Andrew, for a lovely presentation. It was very logical and kind of took through all the different steps. Couple questions. One of the things that I've noticed with the patterns emerging, from Turing systems is that the effective diffusivity or diffusivity has to be quite different. Sometimes order of magnitudes different, and sometimes that doesn't seem quite practical considering the proteins, in reality are very similarly sized. So I just wanted to get your comment on that. The second is a more naive question. I loved your 3-dimensional shapes where you've done the patterning. And I noticed that at the core of those shapes, your activators are essentially, very, very low. and you only see patterns on the surface. Can you comment on what would that mean, kind of physiologically? What does that reveal about the biology, that the center or the core of the structure almost has no activator, but you do see patterns at the edge or at the surface?
Right. So the first question, I should have actually talked about this a bit more. This is a big area with a lot of people talking about robustness of Turing patterns, right? And if there's sort of large Turing spaces in this sort of work. The classical one classical objection to these is that you absolutely need differential transport, right? You either need to think of morphogens which don't move along with diffusible ones, or you need to think of morphogens which move at different rates, which is not, as you say, terribly plausible, given that the molecules are all approximately the same size. We think most of the physical transport approximately occurs at the same rate. So the way people tend to think of this is again that these are caricatures, and you do have proteins, you do have signaling molecules which act intracellularly, and so or in a membrane-bound way, and so they do have different transport properties than other morphogens, right? So in this bacterial work, especially in these synthetic systems, we can characterize all of these diffusion rates pretty easily. The systems are simple enough to actually measure them. And there you very much exploit the idea of intracellular signaling versus extracellular signaling in order to drive this kind of differential transport. There are mechanisms via lateral inhibition that do something similar, right? If you have gap junction transport, or you have various other mechanisms. So there are ways to get around this, but you're right, it's not, this hasn't been, in my mind, fully resolved. And it's related to the fact that there has been work by Michael Stumpf and James Green and quite a lot of other people on the robustness of Turing spaces, and how, if you think of the sort of parameters in which you have these patterns, these sets of parameters tend to be reasonably small in some sense. And real biology is incredibly robust, right? If you perturb a mouse, okay, you may change the pattern if you picked the right genetic perturbation, but in general, you're still fairly close unless you perturb quite a lot, right? Unless you really upregulate or downregulate something. And this robustness is quite strong, and I think our models have not fully explained this, why the model is in some sense more fragile. Some of this is by adding bells and whistles, adding other bits of well, okay, maybe this bit of physics is relevant, or this bit of biology, but I think it's not fully explained. With respect to the core of the pattern, here you mean on the surfaces, you were saying that.
Yeah, so you're, if you look at the toroids or the spheres that you showed with patterns, I could notice that, on the inside, they were entirely blue, dark blue, but on the surface, you could see the patterns. And I just wondered, what does that tell you about that? I mean, it's not plausible that you will have no activator in the inside, right? No morphogen on the inside. So what does that tell us about the biology? That these patterns only kind of show up. The brightest kind of morphogen spots end up on the surface, but in the core, there is nothing.
Yeah, so I should say in the simulations I showed on the tori and the sphere, I actually don't resolve the inside. I'm only thinking of a two-dimensional reaction diffusion system.
I see.
But you're right that you do observe things like what you're saying because of boundary effects, right? We did some work at this, and there's a recent paper out of Sheffield in neuroscience talking about these sort of boundary-driven dynamics. I cannot remember the authors, but anyway, if you email me, I can send it to you. The sort of bound of the region can have an influence. And one unfortunate thing is that fully understanding that doesn't come from the mathematics, right? It comes from it doesn't come from the simple mathematics, the sort of linear stability thing. You need to understand the sort of non-linearities of the pattern, right? The geometry can have roles, even though I've sort of swept it under the rug and said it appears as a single parameter, particularly boundaries, but other aspects of geometry can play important roles. And we think that this is true, right? We think that this occurs in physiology. I mean, you clearly have cell fate boundaries that spontaneously emerge. I shouldn't say spontaneously, but they are there, right? We can observe tissue stratification things.
Thanks. I will reach out, and thanks for an insightful talk. Cheers.
Thanks very much.
Andrew, I just want to say that if you do remember the paper, I already started putting some of your papers in the paper sheet in our.
Okay, sure.
So if you remember other ones, I just kind of typed them. I don't know how all the names that you had there. So if you don't mind, like you can add them and others can look at them too.
Yeah, I can do that. Is this on the Slack?
No, it's the Google shared sheet.
Okay, the one you shared in
the paper
Yeah.
list is super helpful, by the way. I love that.
Okay, great. I just try to keep them like, so I already started one for May 12th and I put, I think one of your first papers in the EMBO in Nature, and I wrote the books, but if you don't mind, like clarifying them or add other ones that you think are helpful, and
Sure.
then people can look at it. Just hoping it helps. Get
I
more-
know we're over time, but can I throw another question in?
Yeah, it's yours. I'm good.
Thank you, Andrew. That was just a fantastic talk. And I really, there was some beautiful math there, and I really appreciate the comments that you had about sort of the human part of how we do what we do. I think it's important for us to be thinking about those things. My question is, so for those of us who haven't worked in these areas, but I work in, models for highly structured and highly patterned sort of systems, but I've never looked at my types of systems in terms of these, possibility of them displaying these types of Turing behaviors. What would inform me as a sort of a mathematically savvy, person working in a system, whether the spatial patterning of the types of systems that I approach, would be amenable to these types of perspectives and models, and whether they would be informative for the types of things that that I do.
Yeah, so this is a good question. Sort of on a tangent, but I hope you'll find this meaningful. Because this analysis is technical enough but simple enough that most people with mathematical training can do this kind of analysis, there were quite a sequence of papers applying these ideas to things like oncology and applying these things to things like developmental biology and wound healing and these other areas. So Murray's book, right, Murray's Mathematical Biology, the second volume in particular, mentions quite a lot of these studies, and often you have a sort of pattern formation analysis, whether or not you expect a sort of periodic spatial pattern to occur on, say, the surface of a tumor, right? And so this is a good and a bad thing. It's a good thing because I think the actual analysis and the actual details of doing it is not terribly hard, right? It's exactly the recipe I showed you for spatially and state continuous things, right? If you have cellular automata, there are a couple extra details, right? There are some other bits, but generally speaking, the procedure is, I think, fairly straightforward and fairly standardized. But there is always this question of this tends to refer to a particular kind of mechanism. It tends to refer to a particular kind of, right? We use this word instability. And this instability has it has a mechanistic or physical meaning, right? It has this meaning that in the absence of diffusion or in the absence of spatial transport, I assume my system is stationary, stable. But when I include the spatial effects, things become unstable. And this has an interpretation that leads to short-range activation, long-range inhibition, I think quite generally. But is that the actual mechanism, right? Because you can also get spatial structure via pre-patterning. You can get essentially whatever spatial structure you want from pre-patterning. One of the troubles is that you can't easily disentangle these two phenomena unless you have a nice separation of timescales in the system, right? So I guess my advice would be to sort of look at Murray's book. I think he presents the sort of ideas of the methods in a straightforward way. I mean, I also have lecture notes that do this, I think, for a fairly large class of systems. Or if you look at the review paper that I mentioned at the beginning, we sort of walk through what the general procedure is.
That's
the
super helpful.
interpretation-
Thank you. I
Yeah.
mean, it's just incredible, you know, to kind of come out of a talk and kind of go, hey, could I do this? And the types of things I work on. I do a lot of stuff with pattern formation for, I work in crystallization. And so, aggregation of proteins and these types of things into structured entities. And so this is great. And I feel real inspired to go and check this out more. So thank you.
Yeah, absolutely.
I had some questions if we still have time. Okay. Yeah, thanks for the nice talk. I really liked your ending too, the thoughtful comments. So this PDEs are not my area. I like to think of it more in terms of like ordinary differential equations. So I'm kind of, I have probably naive questions. At one point you had like these bespoke systems. Were you saying that you can somehow encode arbitrary long time states in the system by choosing the reactions or something, or can you elaborate?
That's essentially it. Yes. So I choose the reactions to vary in space, right?
Yep.
In just a particular way, I think in a fairly intuitive or obvious way. And then when I run a simulation, you see the system fall, roughly speaking, locally into whatever state you encoded.
And in terms of this encoding, is there multi-stability allowed? Can you encode arbitrarily many states?
Yeah, that's a good point. So here we chose a system which I think if I remember the paper correctly, it has two stable homogeneous equilibria. One stable homogeneous equilibria. But when you cross the boundary of the Turing instability, you lose one of these. And so essentially the system is, loosely speaking, singly stable, where it's either going to be a pattern or it's going to go to this heterogeneous steady state. There are hard questions about multi-stability, both in terms of kinetic steady states, but also in terms of Turing patterns themselves tend to fall into a class of different. You tend to get many different patterns for the same system, right?
Okay, so is this like sort of an open question in terms of this class of PDEs? Like their long-time behavior is not well characterized?
I think so. I think that's fair to say. I mean, it's true for ordinary differential equations. When you get to algebraic complexity, when you're thinking of even polynomials, even mass action kinetics, where you don't have detailed balance, right? Then you can typically, it becomes very difficult to bound the number of steady states that you get. And in these PDE systems, that's more difficult because you're not just, I mean, you can't count, right? If you have a system of algebraic equations, there's a lot of machinery from algebraic geometry and things that you can use to count the number of steady states. And so there's a lot of work in algebraic systems biology doing this sort of thing. So Heather Harrington and several other people, I think Michael Stumpf as well, look at this for these, say, large gene regulatory networks. But for Turing patterns, it's messier because the spatial part is harder. It's harder to characterize these inhomogeneous equilibria. They're not simple solutions to algebraic equations, if that makes sense, if that gives you any.
No, it certainly does. Do you have any like leads on like where the, I guess, frontiers of that, like any references for long time steady state characterizations for these?
I wrote some blog posts a few years ago that I'm happy to send. Yeah. I mean, you can make some statements. As I said, you can prove rigorous theorems in the case of a single chemical species. You can completely characterize the equilibria in that case. Mm-hmm. You have transient phenomena like Ostwald ripening or coarsening, if you've seen this. But the steady states, the long time behavior, I think is reasonably easy to characterize. But for systems, right, there's a technical reason you don't have something called a comparison principle, and so that means you can have an explosion, an exponential explosion in the number of steady states. You also have what are called metastable equilibria, if you're familiar with this. So where the system sort of like settles on something, and it looks like it's a stable state, but it isn't. It eventually moves away, and this can take a long time. Anyway, this is all a real pain, right? But it means that people like me have plenty of work to do. Right?
No, that's great. Thank you for your perspectives.
Thank you, Andrew. This has been wonderful. I think we all learned, and thank you for a great talk. We are a little over time, and I think there's a few of us who have to run to the next meetings. I can make you a co-host and you guys can keep going, or I mean, we can resume in a couple of weeks together if nobody has any more questions. Everybody's good. Well, thank you. This is great. And I hope you'll add more of these papers that everybody's asking about, if you don't mind. We'd appreciate it. And
Yeah, I can do that. I'm also happy to respond to emails and things, right?
yeah, so in the schedule, I think I have your email, so people can email you. So if that's, and I think that seems to be happening a lot more within this group. So, but yeah, so the paper also just will help others to link for them if you don't mind.
Yes, I can do that. Absolutely. Thank you again very much for the invitation.
Thank you for